let's understand Celvin's cycle or dark reaction. In this case, the first step that occurs is RUBP coming into the cycle. RUBP, which is ribulose biophosphate. Ribulose biophosphate is a 5-carbon sugar. The structure given here 1, 2, 3, 4, 5 carbon sugar, and it's a ketose. It has got ketone. All right. Now 6 molecules of this 5-carbon gets into the cycle. Rubisco, which is a protein, catalyzes carbon fixation. So 5-carbon molecule gets converted to 6-carbon molecule via carbon fixation. All right. So 6 molecules of carbon are fixed to 6 molecules of RUBP to get 6 molecules of 6 carbon. One which was 5 carbon now gets converted to 6 carbon molecule. But it's highly unstable. So it breaks down to form 3-phosphoglycerate. 3-phosphoglycerate. 1, 2, 3. Phosphate attached at 3rd carbon. So it is 3-phosphoglycerate. Now this is the first C3 compound in C3 pathway. So obviously, you know why we call it as C3 pathway, because first stable compound that forms is a C3 compound. So we call it as C3 pathway. All right. Obviously, this was 6-carbon molecule breaks up into 3-carbon molecule. 3-carbon molecule. So obviously 6 molecules gets converted to 12 molecule. Now what happens here is this which was 3-phosphoglycerate now gets converted to 1,3-diphosphate glycerate. So obviously one phosphate has to come in. From where does it come in? It comes in from ATP. Now since 12 molecules of this molecule is there, so it takes 12 molecules of ATP which gets converted to ADP and inorganic phosphate which gets attached to this part. So we end up with 1,3-diphosphoglycerate. Obviously 12 molecules and still it's 3-carbon molecule. Alright, so phosphate attached over here, phosphate attached over here to glycerate. Alright, now what happens next? Now next is it forms 3-phosphoglyceraldehyde. Now we all know this was acid part. We all know acid gets converted to aldehyde on reduction. So we need hydrogen. So we have got here NADPH to provide hydrogen to form NADP+. Alright, obviously 12 molecules. So we require 12 molecules of NADPH for reduction of acid to aldehyde of 12 molecules. Alright, so fine. So we get in here. So what we end up with is, it is 12 molecules, 3 carbon, but it's an aldehyde now. Alright, so what we saw, here we saw monophosphate, here we saw diphosphate, here it was acid, then this acid got reduced to aldehyde. So 1,3-diphosphoglycerate gets converted to 3-phosphoglyceraldehyde and hydrogenation occurs via NADPH. NADPH gets converted to NADP and it provides hydrogen to the acid to get converted to aldehyde. Alright, so this is the molecule of 3,3-phosphoglyceraldehyde, aldehyde group. Alright, now so what next? So obviously now this comes out of the cycle and it moves ahead. So this moves ahead. Now this moves ahead to form glucose. Now mind you, till now we had 12 molecules over here. Out of those 12 molecules, it's only 2 molecules which are moving ahead, whereas 10 molecules which are there, they are moving back into the cycle. Alright, so we would pick this up later. Let's understand what's the fate of those 10 3-phosphoglyceraldehyde molecules. So this moves ahead to form xylulose 5-phosphate. This is the structure. Now as the structure says, it's a 5-carbon sugar and a ketone group and 1 phosphate. So it is xylulose 5-phosphate. Mind you, it is the D isomer. Alright, D optical isomer. Alright, so xylulose gets converted to erythrose 5-phosphate. So this was 5-carbon sugar and this is 4-carbon molecule. Alright, so erythrose, this is the structure. This is the structure of erythrose. This structure actually I have given because this may come in chemistry also. So erythrose, we have 2 OH this side. Alright, so we call it as erythrose on same side. When we have 2 OH on the same side, we call it as erythrose, but if one is on that side and one is on this side, OH, then we call it as triose. Alright, so anyways, this is D-erythrose because we know that this OH is towards the right side as per L-glyceraldehyde, which I have explained earlier in my earlier videos. So this is D-erythrose, which is moving forward. Alright, so D-erythrose, which has got phosphated in this position, so it is in the 4th position. It has got phosphated in its 4th position. So 4th position, so it is erythrose 4-phosphate, which now see this was a ketose, this is an aldose, this was a ketose, this is an aldose, back to ketose, pseudoheptulose 7-phosphate. Now it becomes 7-carbon molecule. From 4-carbon molecule gets to many complicated reaction, it gets into 7-carbon molecule known as pseudoheptulose 7-phosphate, which then gets converted to ribose 5-phosphate. Now this is 5-carbon molecule. This is the structure 1, 2, 3, 4, 5-carbon, and the 5th position we have got phosphate group attached there, and it's an aldehyde group at the top, 1st carbon, right? So from this it then again moves into RUMP. Now this was ribose, this is ribulose monophosphate. Now this is the structure over here given for ribulose. So ribulose again is 1, 2, 3, 4, 5-carbon sugar and it's a ketose. Alright, and again it is D1, right? D. So phosphate at this position. So this is what is ribulose monophosphate. Alright, so ribulose monophosphate then again gets converted to ribulose biophosphate. Obviously it would be taking ATP. Since we are talking about 6 molecules, so it would consume 6 molecules of ATP to phosphorylate 6 molecules of 6-RUMP to 6-RUBP. So MP and this is BP, biophosphate, monophosphate. Alright, and then again it comes back to the cycle. So we have completed this cycle. Alright, so this is how the cycle goes on. But we did not see glucose anywhere. Okay, synthesis of. So let's now move on to synthesis of glucose. Alright.